Hello guys, my name is Mark Munyi and Chris and I will be presenting about super resolved fluorescence microscopy. So this is a topic that won the 2014 Nobel Prize in Chemistry, where three gentlemen, Eric Beitzig, Stefan Hell and William Mona, were awarded for contribution and advancement in super resolved fluorescence microscopy. Now, for us to understand what this means, we actually need to understand why we need microscopy and some other key factors to consider in microscopy. So microscopy is the ability to view microscopic organisms or very, very small objects by enlarging them. And this is very useful in studying like the internal workings of a cell, and which is in turn helpful for studying how diseases form and things like, drug design and such. And the earliest documented forms of microscopy dates back to around 1000 AD, but the first real microscopic attempt was around 1600 when a microscope was used to study microorganisms. Now, there are three main factors that determine that are very key when doing microscopic imagery, and one of those is contrast, the other is magnification, and the other is resolution. And contrast is just the difference in intensity between an object and its background. So as you can see right here, if this is the normal image, which is like normal contrast, then this is low contrast. As you can see, there isn't really a large difference between the subject, which is this cartoon, and the background. Well, this is very high contrast because you can see the difference in intensity between the foreground and or rather between the object and the background is quite intense. And so this is a very high contrast. So when we are viewing microscopic objects, we need as much contrast or as comfortable contrast as possible so that we can be able to differentiate the object from the background. The other factor, as I said, is resolution. And resolution is the ability to distinguish fine details of an object. Or you can think of it as how different or how neighboring pixels appear. How sharp are they? So as you can see, this is a very high resolution image. You can be able to tell apart all the neighboring objects very clearly, while this one is a very low resolution image because it all looks like one subject. You can't really tell them apart. And so in microscopic imagery, you need as high resolution as possible because that's the only way to tell apart very closely neighboring objects, as shown in this. So this is a very high resolution image. It's very sharp. You can tell apart the details, while this is a low resolution image. And the other factor is magnification. And magnification is just how enlarged an image of an object appears relative to the object. So, or rather, the scaling up. So when you're doing object microscope, or when you're doing microscopy, then you need as large magnification as possible because again you're dealing with a very small subject. So the bigger it appears, the better. So microscopy, the microscopy we are talking about in this case is optical, which means it's using properties of light. And one of the properties of light is diffraction, which is just the splitting of light or the bending of light as it's passing through a narrow slit. And so shown on the left right here is just normal optical microscope, and the highlighted area is the objective lens. And what happens is the object you're viewing, which is right here, acts as a point source, and so light from that point diffracts upwards towards the eyepiece. Now, there can only be so large of a cone, this cone shown right here, before you start getting diffraction. And once you get diffraction, it creates a diffraction pattern, which basically is just creates rings of of high resolution and low resolution on the eyepiece. And so NA is the numerical aperture, which is defined as just n sine theta, where theta is the angle between the normal and this side of the cone, while n is the refractive index of the material. And what happens is you can determine how big of a diameter of an object you view just by adjusting the numerical aperture. Now, one of the earliest researches on microscopy by Abe concluded that you could not resolve anything that had a diameter larger, or rather smaller, than the wavelength of light divided by the numerical aperture. And this was a limiting factor that actually the guys who won the Nobel Prize, Stefan Hell and co., they were able to avoid this barrier. They were able to view objects that were much, much smaller than half the diameter, or rather half the wavelength of light, which is known as the Abbe's diffraction limit. So, all right. So, because you're viewing a very small subject, you need a way of telling it apart from the neighboring objects. For example, think of a case where you're trying to view a mitochondria, for example. Well, you need a way of telling it apart from all the other contents of the cell. And that is commonly done by incorporating fluorophores. Now, fluorophores are fluorescent subjects that you can attach or that can bind to non-fluorescent objects under study, and that makes them glow or more visible under the microscope. And a very good example right here. Assume this is the inner cell we are looking at, and we are trying to look at the mitochondria in this case. Well, you can't really tell which the mitochondria is because it's not tagged. But in this image, you can easily tell this is the mitochondria because it's clearly tagged, and you can see, okay, this is what I'm looking at. And so that's how tagging comes into play. So this is a whole scientific research area with a lot of developments. And so a lot of, tagging techniques have been developed, such as synthetic dyes and antibody taggings. Like shown on the left is a synthetic dye, which basically just glows and it's gonna bind to whatever you're trying to look at. And on the right here is antibody tagging, where you have this antibody that is fluorescent that can bind to whatever the subject antibody you're trying to look at. But one common thing among all of the tagging techniques is breaching, which is the fluorescent dyes or the fluorescent fluorophores will decrease in their fluorescence with time. So as shown in this diagram, if this is what the fluorescence looks like at 0 seconds, well, as time goes by, it's going to decrease and the fluorescence decreases, and so you can barely tell apart after like 15 seconds. And so this is a core part that also those guys were able to avoid by the development of STED that Chris will talk about now.
All right, thank you, Mike, for all of that introduction, which will be really important when we start diving into specifics on how STED microscopy work. But first, let's look at the three awardees and understand sort of the context of why there's three people awarded for this award, right? And so the three awardees were Betzig, Hell, and Moriner. And so Betzig was essentially the first person who said, oh, fluorophores can help look beyond the diffraction limit and put like a label or pinpoint to this specific particle at this specific location. And Moriner took this further and actually studied fluorophores that they could control with lasers. So not something that's just like turned on and off whenever it felt like it, but could be controlled by laser excitation. Hell combined these concepts and created what essentially is the STED microscopy that won the Nobel Prize. So if we take a look at closer how this works. So first of all, a sample has fluorophores added to it and is then excited with an excitation laser. And so this excitation is then hit with a second laser that bleaches the surrounding area except for a small circle. Think of it as a big donut with a hole in the middle. And so once that fluorophores around are bleached, remember, that means they aren't fluorescing anymore. So essentially all you have left fluorescing is this small pinpoint in the middle that is smaller than the diffraction limit we see with like optical microscopes. And so you can see how much resolution, contrast, and like better clarity insight you get into these microscopes. And so on the left-hand side you see here is a conventional microscope using just ordinary fluorophores, whereas STED using the bleaching process can give you a finer boundary for this mitochondria cell in this paper published right here. So, all right. So let's take a closer look at the Jablonski diagrams of the electrons within STED. So let's take a look at what happens with the electrons by bleaching. Like, what exactly is happening? So what happens is that all of the electrons are excited, but the ones hit by the second beam are forced to are de-excited and go back to ground state. And when that happens, they are turned off. They no longer fluoresce because fluorescence is caused by decay from S1 to S0. Right, correct? And so if we take a look at the middle unimpacted beam, we can see that this natural fluorescence occurs. and is a result of just letting it happen naturally. You're not putting any impetus in this. All you're doing is allowing the fluorophores to glow and then release their electrons naturally. All right, so now that we understand how STED works in like a general sense, we can look at how this donut beam impacts the resolution limit. And so in the original Abe's resolution limit, we can see that with STED, we can incorporate an intensity factor for the wavelength minimum, meaning that we can control how like refined we want this data to get. And if we look at this graph here, we can see that this original non-STED based microscopy is so much larger than the 97 nanometer that we get. That gives us a much crisper, more defined pinpoint on like where this fluorophore is. And so this is called a point spread function. And essentially tells you, oh, this is where we think that a fluorophore is. And having a like much skinnier PSF function and compared to this PSF allows us to have a better defined, better resolution, better clarity, better contrast, all that stuff we heard about in the beginning from Mark. And so instead, another pro of this process is that it's a relatively quick process. And so having a quicker process means that we will avoid bleaching, and then we can also get more data points from the same set of data than we would with other methods. And so as we can see here, a regular sampling will get a good amount of data. It's not junk, but instead you can get a larger amount of data, which will give you more details where you can zoom in and see like, oh, this protein is moving through the membrane, or the interior of this mitochondria is changing in respect to this factor, right? And so being able to get that data much faster will also allow you to image live cells, and is really important to see these active processes happening in real time. And so that's actually what they did with this paper. And so what they did in this is they essentially cut out a hole into the mouse's skull and looked at the neurons firing inside of their brain. And they were actually able to image the neuron chains. And right here, you can see the synapses interacting with each other, which is completely like interesting. You can see actual stuff that with your eyes with just the STED method. And so this also has a even smaller 67 nanometer sort of resolution or range with the laser. And you can get really fine details from using STED for biological processes. Some developments with STED over the years has included the 2-channel STED, which is a result of using 2 different fluorophores, which de-excite at different wavelengths, just enough so that you can collect 2 different sets of data within the same cell or same setting. And so there's also a significant resolution increase as a consequence, being able to coordinate with each other and see, oh, this is one thing, this is another thing, instead of having all blur. And so you can get from a 97 nanometer to almost down to a 35 nanometer resolution, which is a really impressive jump. And you're essentially being able to see 2 different systems interact with each other throughout a process. All right, and so some of the work around STED is sort of exploring alternatives or building off that basic principle of how STED microscopy works. And so in this process called ground state depletion, what happens is that instead of going straight down and like de-exciting the electrons from the S1 to S0 state, instead it tries to utilize the intersystem crossing and go into a triplet state and then sort of keep the depleted electrons there before completely depleting them. And so why exactly would that be beneficial in any sense? Well, essentially what happens is that instead of, what usually happens is when you go from S0 to S1, you still have like some residual electrons in that donut beam all around that are not, like still appear in the signal. But with this process, by moving it to a different state or a meta-dark state, you essentially get no signal at all. So you go from having a little background noise to no background noise. And because of this, you can get an even better resolution with the better resolution in the point spread function, which allows you to observe these smaller molecules with even better details. And so future works in microscopy and such will continue looking at different previous pathways, such as D-storm and such, to see how we can better refine what we see inside living cells with fluorescent microscopy.